And now I have the honor of introducing today's speaker. So for those familiar with me, you'll know that I worked for well over 10 years on a book dealing with labor politics and struggles in some of the world's largest industrial pork production facilities. But back in 2014, I was writing my first article, which dealt with pig diseases and viruses passing across workers' bodies. While trying to develop that article, I encountered this path-breaking and inspiring piece that was making waves out of North Carolina. In my own words or phrasing, this research illustrated how factory farm workers' bodies literally carry burdens of animal agribusinesses' efforts to make their hogs grow, I would say, perversely fast. The paper found that laborers took home in their nasal cavity livestock-associated antibiotic-resistant bacteria, and they are carriers for up to 96 hours after they left work. For me, it subtly remade how I thought about my own time physically working in breeding barns, gaining a sense of how different species' industrialized health conditions jump across and blend together in these confined spaces. It added yet another potential dimension to think about in terms of industrial farm workers' vulnerability. And in my opinion, it was a brave piece of scientific research, given how often farm workers are written out and ignored in discussions of animal agriculture. In any case, about a year ago, I remember thinking back to that article and wondering what the lead author was currently working on and where she had ended up. And I was stunned to discover that, in fact, we were at the same university, at Tufts. So Dr. Maya Nadimpalli is a research assistant professor in the Department of Civil and Environmental Engineering here at Tufts. She received her undergraduate degree in environment from McGill University in 2009, and she completed her Ph.D. in environmental sciences and engineering at UNC Chapel Hill in 2015. As we'll learn today, her research combines tools from environmental microbiology, epidemiology, and genomics to understand the consequences of feeding animals large quantities of antibiotics on the selection and spread of emerging resistant pathogens to vulnerable human conditions, both in the United States and across the globe. Please join me in welcoming Dr. Nadimpalli.
Thank you, Alex, for the introduction and for inviting me today. And full disclosure, this is my first in-person seminar since COVID, so FYI. Okay, so, oops. It's going to be a little bit of back and forth here as I try to figure out how to make this work. But so I thought I'd start today by giving everyone a little bit of a primer on what antibiotics are and what resistance is, to make sure we're all starting on the same page before I get into my research. So antibiotics are used to treat bacterial infections. Specifically, they're small molecules, and we use antibiotics to treat a wide range of infections that many of us have had. So things like staph infections, urinary tract infections. Antibiotics are relied on to treat post-surgical infections, and several STDs are caused by bacteria as well, things like gonorrhea and syphilis. We need antibiotics to treat these infections. Now, antibiotics work by either killing bacteria or preventing their growth. And I'm sure everyone knows this, but just to remind you, antibiotics do not work against viruses. There's no point in taking antibiotics if you have the flu or the common cold. So antibiotics, although we consider them to be an essential part of modern medicine, they're actually not that old. We haven't been using them for that long. So antibiotics were accidentally discovered by Alexander Fleming in the 1920s. He was actually, I guess, known for being a bit of a slob in his lab. And so what I'm showing you here is a picture of what's called an agar plate. So this is a plate containing a jelly-like substance packed with nutrients that bacteria really, really like. And so we use agar plates to grow bacteria, which are normally naked to the human eye. We allow them to sort of amplify so we can actually look at them and characterize them. And so Alexander Fleming was trying to grow Staph aureus. And those are the white colonies you see kind of at the bottom of the plate. And these bacteria cause staph infections. And the work that Alex was talking about from North Carolina related to hog production, that was focused on this kind of bacteria. You can see, however, that his plate became contaminated by a fungus at the top. And what he noticed was that, away from the fungus, these Staph aureus bacteria were growing just fine. But as you got closer and closer to the fungus, the bacteria were having a much more difficult time growing. So he was able to isolate the compound that this fungus was producing, and therefore we had penicillin for the first time. And he realized that even when diluted 800 times, this compound was still effective at inhibiting the growth of Staph aureus. So antibiotics, or penicillin specifically, was first given to soldiers in World War II. And by the 1950s, we had figured out a way to begin mass producing this compound so everyday individuals could start using it. Now, antibiotics absolutely revolutionized medicine. And I want to give you some examples of infections that we think nothing of today that were previously very, very difficult to treat or deadly. All of us in this room have at one point or another had strep throat, I'm sure. Prior to the advent of antibiotics, strep throat could sometimes advance into something called scarlet fever, which you may have heard of, and that was highly fatal for young children. Tuberculosis is also caused by a bacteria prior to antibiotics. The best thing you could do was to go to a sanatorium. Some of you, again, might have heard of that term, and that was literally just a place where you were encouraged to rest and take in fresh air. That was the best we could do to try to prevent or reduce your active TB infections. Meningitis is also caused by bacteria. Before enrolling in Tufts, all of you were probably required to show evidence of taking a vaccine against bacterial meningitis, and that's because it is spread really easily in shared housing like dormitories. Meningitis was often fatal prior to antibiotics, or if you recovered, it could cause serious neurological side effects. So I've shown a picture of Helen Keller here, who is a household name because of all the good she did in her life. She, while also being blind and deaf, she was not born that way. She had a severe infection when she was very young. Based on the symptoms, doctors nowadays think it probably was a bacterial meningitis infection that caused her to be deaf and blind. And I've saved probably the most horrifying picture for last. I mentioned syphilis is caused by bacteria. This is a sexually transmitted disease, but in its later stages, it actually attacks organs and can cause the bridge of your nose to collapse. And syphilis is unfortunately also spread from pregnant women to their children. And prior to the advent of antibiotics, mercury was a preferred treatment for curing syphilis infections. So some of the things I'm describing to you probably sound basically medieval, right? But these were some of the only options available to people less than 70 years ago, which is something we don't really think about very often anymore. Now, the fact that we are seeing increasingly bacterial infections that cannot be treated with antibiotics is really undermining modern medicine. So I have a little cartoon here to show you in a very simplified way how this can happen. So let's pretend the gray circles that you're seeing on the screen are some species of bacteria living in your gut. Now, bacteria are constantly multiplying, and as they multiply, they can make little mistakes when they replicate their DNA called mutations. And sometimes these mutations can actually confer a beneficial effect. They can be protective for that bacteria. So let's say the person whose gut these bacteria are living in takes antibiotics. And let's say this bacteria has developed a mutation that has allowed it to encode a thicker cell wall. That's what this purple circle is supposed to represent here. All of a sudden, this mutation is advantageous, right? Because the antibiotic cannot penetrate the bacterial cell wall, and the bacteria can survive in the presence of antibiotics. And not only can it survive, it's going to multiply. Bacteria also have the ability to basically pass the blueprints for resistance mechanisms between each other. They can kind of share that knowledge between them. So here we have a different kind of bacteria in brown. And this resistant bacteria is able to share a blueprint for encoding a thicker cell wall with this other strain. And now we have multiple strains of resistant bacteria living in an individual's gut. And this is, again, just a cartoon, but is an idea of what really happens. So antibiotic resistance has continually increased for many infections, many very common infections. And so, we see multi-drug resistant gonorrhea, drug-resistant UTIs, skin infections, hospital-acquired infections. And antibiotic-resistant infections are concerning for multiple reasons. So first, if you have a resistant infection, assuming we have an antibiotic available to treat that infection, that antibiotic can have much worse side effects and be much more expensive to use, right? So for example, some of you might have heard of colistin. This is considered a last resort antibiotic, but it can cause kidney failure. So maybe we can use it to cure your bacterial infection, but we really wouldn't want to be using colistin if we could avoid it. And then, of course, we see infections in clinical settings, sometimes in the US and quite often in other parts of the world, that are not treatable with any available antibiotic. And the concern is that as more drugs stop working, more lives are going to be put in danger. And so, maybe you all have seen headlines like this in the news. So there's a lot of concern about the growing threat of antibiotic resistance. We see headlines like, "Are you ready for a world without antibiotics?" And the idea that the world is headed for a post-antibiotic era. And these are all headlines I pulled pre-COVID. So of course, COVID kind of exacerbated a lot of these concerns because especially early in the pandemic, when doctors weren't sure how to treat patients, there was a huge spike in antibiotic use. So in the middle, there's a CDC study that said, compared with historic levels, increases in patients dispensed azithromycin, which is a very important antibiotic. Prescriptions were highest in New York and New Jersey in March and April, which coincides with the start of the pandemic when there was really a scramble for what we could do to help save people's lives. Now, from what I've told you so far, you might think that resistance is primarily a clinical problem and that it's coming about as a result of the overuse or misuse of antibiotics in human medicine. But actually, most antibiotics that are sold across the globe are for use in animals, not for use in humans. To give you a sense of perspective, almost 75% of antibiotics that were sold in the US in 2016 were for use in animals, not for use in humans. And for the most part, it's not animals raised in these kinds of settings that are consuming antibiotics. So animals raised outdoors and on pasture, those are not the main consumers. And instead, it's animals raised in settings that look like this, or like this, operations that are often called confined animal feeding operations here in the United States. Antibiotics are used for three main purposes in food animals. The first is to treat disease. So just as you and I would take an antibiotic if we were sick, if an animal is sick, we can give them antibiotics. Antibiotics are also used prophylactically. So let's say one animal in a barn of hundreds or thousands of animals becomes sick. In order to prevent that infection from spreading, farmers and vets will actually just give antibiotics to all animals inside the barn. And then a third reason that we use antibiotics, and for years this was the reason why we use most antibiotics in animals, is to promote growth. So as Alex mentioned in his introduction, if you give animals a small dose of antibiotics every single day through their feed or through their water, it actually helps them gain weight more quickly with the same amount of food, which means that you can slaughter animals more quickly. You can bring in a herd more quickly and therefore increase your profits. And so I do want to mention that since 2017, medically important antibiotics, so antibiotics that are important for us, for infections that we have, they can no longer be used for growth promotion, but that was only 4 years ago. They were used until then in the United States. And the concern here is that the process of resistance being selected for in the gut of a human that I described to you earlier, that exact same process can happen in animals. And so there are, when something like that happens, it's possible for resistant bacteria that are selected for in animals to spread to humans and vice versa as well. So one way that resistant bacteria could spread to humans from animals is through direct contact. And so that is how I actually started working on this topic through my work with farm workers in North Carolina. And so just as Alex mentioned, we were finding that workers on hog operations in North Carolina, their family members were being exposed to multi-drug resistant staph that we demonstrated was originating in animals. And being colonized with these strains does increase your risk for subsequent infection. Now, the other way to potentially be exposed to these bacteria is through contaminated food, theoretically. However, in the United States, our food system is generally very safe, right? So certainly we do have foodborne outbreaks once in a while, but for the most part, we have laws and regulations and infrastructure at nearly every point along the food supply chain to make sure that even if we do have resistant bacteria being selected for in animals, the average consumer, here in Medford, Massachusetts, is not going to be exposed to these strains. Now, that is not the case in many countries where this type of production model is becoming increasingly common. And so there is an increased demand for animal-based protein all across the world right now, as I'm sure many of you know. And that's in part due to the country's development and middle classes expanding. And certainly the increase in demand for meat is a result of wanting to improve nutrition, but also it has a large part to do with the increase in fast food and this idea that being able to eat meat every single day, maybe multiple times a day, is a symbol of prosperity and wealth and Western capitalism. I'm going to be talking about antibiotic resistance in my talk, but as I'm sure you all are aware of, this dietary transition in general has a lot of other implications for human health. So we see increased risks for obesity, cardiovascular disease, diabetes, and of course, animal production produces a lot of greenhouse gases. And so there's major implications for climate change as well. So I'm just going to talk about one little part of the impact of this transition on planetary health, but just keep in mind there's a lot more going on there as well. And so the types of farms in general that are proliferating across the world in order to meet this increased demand for meat are these kinds of large-scale farms that I was just describing to you, which really rely on antibiotics to promote growth, but also to prevent and to treat disease. So as a result of these changes, it's estimated that compared to 2015 levels, by 2030, there's going to be nearly a 70% increase in antimicrobial consumption among livestock. And the countries that are projected to have the greatest percentage increases are all low and middle income countries. In fact, it's expected that animals raised in China alone will consume 1/3 of all antibiotics sold across the globe for use in food animals. Now, the reason that I started becoming really concerned about this is because those public health protections that I was describing that here in the United States limit bacterial exchange between food animals and the general population. So, things like our safe food supply chain, but also basic things that we take for granted, like access to clean drinking water 24 hours a day. Most of us have access to flush toilets. We have centralized sewage treatment. And we have regulations which aren't always enforced, at least in North Carolina, but generally we have regulations to prevent untreated animal waste from being dumped directly into surface waters. Now, many of these public health protections do not exist in other countries. So either the regulations don't exist or the infrastructure doesn't exist. And so I have a visual to help you kind of think about the potential implications of this when it comes to resistance. I know this figure is a bit complicated, so I'm going to walk you through it. So first, as I mentioned, intensive farms, and by that I mean farms where you have hundreds, sometimes thousands of animals raised in confinement, these types of farms are becoming more common in many parts of the world. And the issue is that waste generated from these farms, as I mentioned, there might not be any regulations in place, or if they're in place, they're probably not enforced, regarding how this waste should be treated before it's disposed of in the environment. Now, that's concerning in places where a substantial portion of the population might be relying on untreated water as their primary source of drinking water, or if that untreated water is being used to irrigate crops like produce. Now, in many settings, in Cambodia, for example, where I'm going to describe some work that I've been doing for you, although intensive farms are becoming more common, most animals, 80% of animals in Cambodia, are actually still raised in households, so in small-scale household settings. There are many potential opportunities for pathogen exchange between humans and animals in settings like this, but I just want to point out one of them to you. So in rural Cambodia, 30% of the population still practices open defecation. Now, animals that are raised on family households, they're not penned up for the most part. They're allowed to roam freely during the day, which means that there are opportunities for animals to come into contact with human feces and even consume it. And so this is a situation where drug-resistant bacteria or pathogens in general that might be circulating in humans could spread to animals. And I point that out because that is a direction of transmission that we never even think about in the United States. In addition to places where animals are raised, we also have fresh food markets, sometimes called wet markets, which are really important spaces for access to fresh, affordable food. However, food safety and hygiene regulations, if they do exist, aren't necessarily always enforced. And the last thing I want to point out here is that in many countries, antibiotic use is not well regulated, meaning you do not need a doctor's prescription in order to purchase antibiotics for either yourself or for the animals you're raising. So this is a kind of situation that results in us seeing oftentimes the same antibiotics being used both in humans and in animals. So I am going to now describe to you a couple of case studies that describe some work I've been involved in. After I did my PhD in North Carolina, I was lucky enough actually to go to the Pasteur Institute in France for a few years. And I joined a team there that was leading a multi-country project called the Birdie Project. And they were interested in understanding the burden of bacterial disease among very young children in several low-income countries. So although the focus of this project was primarily on children's health, they were also collecting some samples from mothers. Specifically, so they were enrolling pregnant moms, and right before these women gave birth, they would collect a stool sample. And they were testing these stool for the presence of a bacteria called ESBL-producing Enterobacteriaceae. Enterobacteriaceae is a family of bacteria that includes Salmonella, E. coli, Klebsiella pneumonia, pathogens you all have probably heard of. ESBL stands for extended spectrum beta-lactamase. This is a gene that if bacteria carry this gene, they're resistant to multiple classes of antibiotics that are super important for treating Enterobacteriaceae infections. And just to impress upon you how dangerous these bacteria are, in 2019, the CDC flagged them as a serious threat to human health. So again, to give you some perspective, in the Paris area, about 5% of healthy people, so they don't have any symptoms, they're just healthy people living in the community, they carry this drug-resistant bacteria in their gut, okay? Through the Birdie program, they were finding that women in Madagascar, about 20% of them were carrying these bacteria. However, in Cambodia, they were finding that 70% of moms were colonized. These are healthy women living in the capital city. And actually, these are data from 2016. A study published this year found that the rates are now closer to 90% in the community, so extremely high. So given the work that I had done for my PhD and my understanding that intensive farming was escalating in Southeast Asia, I hypothesized that maybe meat and fish could be a source of these drug-resistant bacteria to the general community. So we conducted a small market sampling study in Phnom Penh. We visited 2 major markets in the city and collected 150 samples, including fish, chicken, and pork products. And then we used something called whole genome sequencing that would allow us to compare in great detail the bacteria that we were recovering from these food items to the bacteria that we were seeing in healthy women. And we also did some comparisons to infections seen in the hospital setting. The first thing I want to point out is that food samples were very contaminated. So we found that over 60% of the food samples we were collecting had these ESBL producing E. coli on their surfaces. And we found that nearly 1 in 5 samples were contaminated with ESBL producing salmonella. Again, to give you some perspective, here in the United States, we have a program called NARMS. I forget exactly what it stands for, but it's a national surveillance system. And every year they screen hundreds of retail meat samples for the presence of a range of pathogens, including drug-resistant pathogens. And they specifically look for ESBL producing salmonella. Now, when we did this study, at that point in time, NARMS had only ever found 1 sample ever with this bacteria on them. And meanwhile, we found 1 in 5 from just 2 markets in 1 city were contaminated. So by doing whole genome sequencing, we could also look at the specific ESBL gene that I mentioned, look at its sequence. So, in the UK, in other European countries, in the United States as well, we do see these ESBL producing bacteria in products from animals, although at very low levels. And as I mentioned, we also see these bacteria among healthy humans. However, what you can see here is that these populations are essentially distinct. So we do have drug-resistant bacteria in humans. We have drug-resistant bacteria selected for in animals, but there's very little overlap. They're circulating kind of independently. However, in Cambodia, we saw essentially complete mixing. So we saw the same genes circulating in humans as we saw in animal products. And maybe that finding is not so surprising when you think about all the possible transmission routes for pathogens in general, but drug-resistant pathogens as well, between humans, animals, and the environment in settings like this. But again, keep in mind that these are the kinds of settings where intensive animal production is increasing. And at this point in time, we really lack human, animal, and environmental health data from many low and middle income countries where this practice is becoming more common. So second, I'm going to describe in very little detail, because this work is ongoing and hasn't been peer reviewed or published, some ongoing work right now in Lima, Peru. So Peru is one of the fastest developing countries in Latin America. It's beautiful, it has great food, also a place where chicken production is rapidly increasing. So I'm doing this work in collaboration with Johns Hopkins, Cayetano based in Lima, and CTSI here at Tufts. And we're leveraging samples that were collected in an urban shanty town of Lima. Shanty towns are often referred to also as slums or urban informal settlements. These are places where the government is not recognizing people's legal rights to live where they're living. And as a result, there is just generally not very good public health infrastructure, right? So people might not have access to clean water. They might not have access to toilets. We know that food safety and hygiene is particularly poor in these settings. They're crowded. There's lots of animals, but also vermin like rats and flies in these settings. And basically there's a lot of opportunities for pathogen transmission. And so we're doing a broader study related to children's health and the gut microbiome. But we noticed that these kids are very commonly, again, colonized with these ESBL producing Enterobacteriaceae. And so a collaborator had a collection of strains from whole chickens purchased at markets in the same neighborhoods where kids were living. And so again, we decided to do sort of a similar approach as we did in Cambodia and compare the strains we were seeing in young kids to strains we were seeing in chicken. You can probably guess what we saw. Essentially the same thing. We see substantial overlap in strains and ESBL gene types between children and chickens from markets. We actually see sharing occurring even when a child is exclusively breastfeeding, which was surprising to me. And what that really suggests to me is that chicken consumption is not the only route of exposure to these strains, right? So it suggests that these reservoir resistant bacteria in humans and in animals, they're very intimately linked, probably through massive environmental contamination. So you could imagine that mother's hands or surfaces in the home or children's or water used by the household or soil in the household, these could all be contaminated by bacteria from animals as well. We really don't know what the main routes of exposure are. So I hope I've impressed upon you this idea that antibiotic resistance is really a One Health issue. And this is something that I'm continuously advocating for through my research. And what I'm trying to demonstrate here is that, we're really concerned about antibiotic resistance in humans. I understand that. But if we really want to curb the selection and spread of resistance in humans, we need to start thinking more carefully about the role that animals and the environment are playing in supporting the selection and spread of drug resistance mechanisms. So I wanted to end on a more positive note. I'm not the only person concerned about resistance. There is a lot of global concern about this right now. And I've just listed some of the initiatives that I'm aware of on this screen. There's a lot of money being directed towards research and initiatives to try to combat this problem. And I want to talk to you about work that's going on at Tufts related to this. So here at the bottom, you'll see the name of the Stuart B. Levy Center for the Integrated Management of Antimicrobial Resistance. So as I'm sure you all are aware of, Tufts is kind of in a unique position, right? So we're here on the Medford campus. We have the School of Engineering, we have environmental studies, we have liberal arts, law. We also have a medical campus, and we also have a vet campus out in Grafton, which I personally had never been to, but I heard is very pretty. But what's particular is that we have entire campuses really focused on human health, environmental health, and animal health. And so this really puts Tufts at an interesting position, an interesting vantage point to start thinking about ways to combat resistance from this One Health perspective. And that's actually part of the mission of the center, which was created, I think, right at the end of 2018 or the start of 2019. It's the main kind of center is right now at the medical campus, but it engages faculty from all across Tufts. I'm part of CMAR as well. So part of the mission of CMAR is to integrate solutions across human and veterinary medicine, stewardship, and awareness. And faculty who are involved with CMAR are in engineering, like me, in drug development, epidemiology, global health, environmental surveillance. I've included the website here, and I would encourage any students here or who are listening who are interested in any part of what I've talked about today or concerned about resistance to take a look at all of the faculty at Tufts across all campuses who are involved in some way with research related to antibiotic resistance. I'm sure that many of them would be very happy to have you help work on projects and become more acquainted with this issue. And with that, I'm happy to take questions. Thank you very much.
Hey, can you talk a little bit more about solutions or what's being done moving forward? Because it sounds like there's both the sanitation side and maybe global development side, and then also with antibiotic distribution and where you think the leverage points are, both in antibiotic distribution for human health and also agriculturally.
Yeah, so thanks for bringing that up. So in terms of distribution, one of the concerns about antibiotic resistance is that we have not been developing new antibiotics for a very long time. And there's a lot of efforts right now being put into trying to restart that drug development pipeline. Probably a lot of you are aware of this, but obviously many pharmaceutical companies realized it was a lot more profitable to develop medications for non-communicable disease where you have to take a pill every day versus antibiotics where you take it once and then the infection is over. So there's a lot of work in trying to develop new drugs and therapeutics. From my perspective, that's really important, but that's not going to be enough, right? Because that's not going to address the underlying causes leading to the spread and selection of resistance. So it's interesting because we're at this point right now where we have this huge water and sanitation kind of global health platform, a lot of researchers, a lot of investment in that area, a smaller kind of contingent, I think, focused on food safety and security. And there's just less overlap, I would say, than I would like to see. So, there's a lot of WASH researchers who are trying to think about what is it that we can do to improve sanitation or access to clean water to reduce the spread of resistance. But a lot of that research is focused on the household level and is not considering the animal side at all, especially this industrial farm side. So I think this is a direction people are trying to move in, but we're really not there yet at all.
Hi.
I have a question.
So,
Sorry,
should I go?
please go ahead in person.
In terms of like antibiotic use in like animal agriculture, at least in like the US, do you think there like is a way to like stop or like minimize antibiotic use in the US just due to like the, I guess, priorities of like a lot of these agro food companies, or do you think it's like not possible like within the system that we have now?
So what's interesting for me is that, I wasn't involved in a lot of research saying resistance is bad, it's spreading to people. A lot of companies like McDonald's, for example, has said they're not going to buy meat raised with antibiotics anymore. And that's because a shift in consumer preference. So I think consumer preference is a much bigger and more important driver of policy related to food and animal production than I, previously appreciated. So obviously consumer preference, I think, changed as a result of science and scientific studies, but it wasn't a result of us talking to policymakers. It was by informing the public who then made sure that their voices were heard. So it's kind of a different route than I was expecting.
I have a couple questions coming in online. From Shivani, I have the question, how is the US managing large-scale meat production without the use of antibiotics? Is there an equally harmful substitute that is not regulated? And maybe I'd just ask and tag on to that. I noted that, if I'm not mistaken, your earlier slide on relative consumption of antibiotics between humans and animals was from 2016. And I was curious if we have seen much of a significant decrease in farm animal antibiotic use since use for growth promotion purposes was theoretically outlawed in 2017. Like, have we seen a measurable decrease in farm animal use since then?
Yeah, so thank you for the questions. I'll answer your question first, Alex. So yes, after that ban was put in place, there was a pretty sharp drop in antibiotic sales. But I don't know if you've seen the recent numbers, but they went back up, and I'm not entirely sure why. I used information from 2016 because there's actually it's not very easy to calculate the amount that's used in animals versus humans. It's not like readily available. So the Pew had done this analysis, so I used those numbers, but it's not as clean cut as we would have hoped, I think, after that ban. In terms of other products that are being used maybe in place of antibiotics, so the European Union, as it is in many other situations, was kind of way ahead of us in being concerned about antibiotic use in animal production. And so they've had a lot of efforts to reduce antibiotic use over the last 10, 15 years. A lot of that has been changes in infrastructure in order to just reduce disease in these settings where you have many animals in small spaces. But there was also a switch initially, and I'm not sure if that's happening here in the US, but there was a switch to using metals in feeds. And unfortunately, they realized that just using metals was kind of co-selecting for the same resistance mechanism. So it wasn't really doing the intended job. And I'm actually not sure how they resolved that, but I think it's been more of a focus on infrastructure changes versus trying to find substitutes for antibiotics.
Thank you. I have another question coming in, and we've never tried this, but I'm going to I have a raised hand, and I'm going to call on Aram Sitar to speak.
Yes.
Thanks so much. Yes, Zoom, this works. I actually just wanted to pick up on the point that you had about changes in consumer preferences that are actually having an effect on affecting the food system, right, and what is being produced. So my question is, what are our affordability and equity considerations there? If people who can sort of go to grass-fed, omega-3 rich sort of food, well, that's absolutely wonderful. But for the most part, that's, a smaller segment of the population. So how do we expect that sort of large scale transformation?
Yeah, that's a great question. And, I don't have the expertise to answer that. I hope people are thinking about that. I mean, for me, that's something where you need kind of government level action, right? To make sure that everybody has equitable access to the safest food possible. I don't know how we achieve that in this country, but I think it has to be done at the government level. I agree with you. It shouldn't be up to consumers to have to burden those costs. It's not right. Yeah, I don't know if Alex, you have any thoughts on that actually.
It's a big question, but also I think has to involve efforts to reduce meat consumption, period, especially in this country here. But we'll go on. I think we're out of questions here online for now. If we have any in the room.
I have one more biological question. So it was very concerning to see that such a big proportion of the population had this ESBL producing
ESBL
bacteria. So I was wondering, in the absence of antibiotics, do they thrive? Do they outcompete the regular gut bacteria?
The ESBL producing strains? It's an interesting question. I was just talking to people about this last week. So, generally our understanding of antibiotic resistance is that it's antibiotic use that drives the selection. So theoretically, if you remove the antibiotic selection pressure, over time, at least, these resistance mechanisms should fall away. I was talking to people last week and they were kind of asking more fundamental ecological questions of, okay, antibiotic use is high in Southeast Asia. Why is 90% of the population colonized? That's extremely high. Is there something different about the ecology of their gut microbiomes, of the environment? I think it's an unknown question. So generally, when a bacteria is carrying a resistance gene, that's a cost, a metabolic cost for the bacteria. So unless it's conferring some advantage, the bacteria will drop the gene. I don't know, maybe those genes aren't costly for the bacteria in those settings. And we don't know. I think it's like a very fundamental science question that we don't fully understand.
Right, and that's exactly what I was wondering. Thank you. Other questions here?
I have a question online, if that works, from Christina Munoz. So the question is, so would, in theory then, consuming meat produced in industrialized countries and exported rather than meat which comes from developing countries help to reduce antibiotic resistance?
I'm not sure if I understand the question. Reduce it for who? Just globally?
I just have a question on that. Yes.
Okay.
Yes.
Well, I don't know, based on this very new work from Peru, I think meat consumption is contributing to resistance, but now I'm not as convinced that it's the whole picture. So for example, produce is very heavily contaminated, and the hypothesis is that it's because people are using wastewater from humans or from animal production directly to grow that produce. I think the connections or the spread of resistant bacteria from humans and animals in the environment, I think those networks are a lot more complex than even I was thinking, when I started doing global health work. In terms of like import, export, and those kinds of things, like we have certain regulations for how clean meat should be when we export it, and to on that same token, when we import things, we carefully screen to make sure that they don't contain pathogens, and we screen for a variety of outcomes. So I'm not sure if import-export kind of considerations are going to help the global problem of resistance. I think it's more like local or regional spread and food trade systems that are involved there. Like, for example, if a producer of shrimp in Vietnam wants to export shrimp to the United States, there are very specific regulations that they have to make sure their food conforms to. And I don't think that's the case if they're just shipping it to Thailand or to Cambodia. And so, yeah, basically, I'm not sure that approach will work.
I have another question, maybe a comment from Claire Giddens-Williams. Very interesting. The UK are avoiding landmark legislation to stop prophylactic use of antibiotics, and MPs have just voted against legislation in the Environment Bill in the UK to restrictions on dumping raw sewage into our seas and rivers. The question of antimicrobial resistance is hidden from public understanding. And maybe I just tack on a question of your thoughts, Maya, on prophylactic use. And I guess I'm curious, in practice, is it kind of hard to distinguish between prophylactic use and growth promotion use and so forth?
Yeah, so for sure, you know, I think that was a concern people had with this ban on using antibiotics for growth promotion, which is that people are just going to continue using antibiotics and relabel it as prophylactic use. I think that's certainly a concern. In terms of like banning prophylactic use in general, I think the issue is that obviously when we want to reduce resistance, our gut reaction is like, let's cut down antibiotic use in animal farming. But we need to provide alternatives to farmers who are relying on animal production as their livelihoods, right? So they need to be able to reduce disease in their flocks in a different way. And so, you know, whether that requires government investment in new infrastructure, certainly like in developing countries, we need to provide technical assistance to farmers to help them figure out ways to use locally available materials to reduce the spread of disease. But we can't just say you can't use antibiotics anymore, good luck, because that's going to result in a lot of animals dying, and it's going to result in people losing their livelihoods. And I think that's why there's been this pushback, not just in the UK, but other countries that have tried to curb antibiotic use in animal production.
Thank you so much for your very interesting presentation. I'm Hai from Vietnam. I'm very interested with your topic because you also talk about the developing country mind in mind. I have a question that in your presentation, you also talk about a research about how the antibiotic resistance can affect the health. It's under the One Health discipline. And I want to know more about, do you know any project? It's like intervention project to raise the awareness about the risk of antibiotic resistance. Could you share?
Yeah, so I mean, for example, like the WHO, I believe, sponsors like an antibiotic awareness week. In general, I didn't mention this, but the WHO, FAE, and OIE have banded together and put together a lot of money to help specific countries develop action plans to combat resistance from this One Health perspective. So they've developed action plans for 60, 70, maybe more countries. And now these action plans have to be implemented, which is a whole different story. But I know the WHO, OE, FAO are like actively trying to help governments figure out ways to reduce resistance, and increase awareness is a huge part of that as well. I don't know how effective these things are yet, but there is action in that arena.
Thank you.
Another question here.
So in combating resistance, do you think that the like the path going forward should be an engineering like biotech solution of coming up with better and like rotating antibiotics, or should it be more of a like systematic infrastructure change in changing the way we do like farming and agriculture?
I mean, ideally both. We definitely need new drugs and new therapeutics, hands down, that's for sure. What's concerning for me is that the new drugs and therapeutics that are being developed, they're extremely expensive and they're for extremely specific indications, to the point where if you don't live in a big city in the United States, I don't even know how you're going to be able to access some of these antibiotics at the point we are now. I think what's stymieing kind of this systemic big picture look at how farming and sanitation and all of these things play a role in the spread of resistance is that most research on resistance transmission has been conducted in high income Western settings, right? Where we have like access to water, sanitation, hygiene, where we have food produced separately from human communities. And so, you know, and there's just a lot less research from low and middle income countries. So it's the perspective I get from a lot of people is that, you know, if we're able to reduce antibiotic use by making people have to get prescriptions in order to get antibiotics in low and middle income countries, that's going to be enough. But it's because we've never had to think about the role that farming animals in your house or the role of not having clean water and sanitation might play in the spread of transmission. And that's a major barrier, I think, right now to getting people to change policy.
Thank you.
Mm-hmm.
We have one final wrap-up question from the audience, from Shivani Desai. What kinds of opportunities could one get involved in at CMAR, and what kinds of skills would you look for?
Yeah, so I think it depends on, so the way CMAR works right now. So first of all, I would encourage you to reach out to Rima. She's kind of the admin for CMAR and she can provide you with more information. I think in terms of skills and opportunities, that depends on what kind of faculty member is doing work that you find most interesting. So for example, there are a lot of people at CMAR doing drug development for sure. There's also a lot of people in the Center for Science Education trying to develop curriculum and training activities to help students from the high school through college age try to understand the connections between humans, animals, and the environment and resistance. And so there's these pillars of education, drug development, surveillance is a big thing, particularly among clinicians. I would say, you know, what I do is also part of CMAR, but I'm leaving Tufts in the spring, so unfortunately that's not going to work. But I would say reach out and take a look at what faculty that you're interested in are doing. It's going to vary widely depending on what you're interested in.
Thank you so much. I'm going to ask Dr. Gomez in the room to close us out because I have yard work happening outside my house and it's very loud.
I think they are everywhere today. We have them too here outside the room. But thank you so much, Dr. Nadimpalli. This was super informative and I wish you the best in your new destination.
Thank you. Thanks for having me.
And please join us next week on a talk on agroecology from actually one former EMBS alum. She's coming back to, well, virtually back to Tufts. So I hope to see you next week. Bye, everybody.
How do I turn this off?